Good evening, everyone. My name is Monojit Assistant Professor, Department of Physiotherapy, University of Science and Technology, Meghalaya. Today, I am going to talk on the topic called staining techniques in microbiology. So, first, let's start with introduction. So, bacteria are the microscopic organisms. They are colorless most of their part. In order to visualize them, to study their structure, shape, other cultural characteristics, it's necessary to make them more easily visible. This means the structure have to be contrast from the environment so that they can be easily seen under the microscope. So staining is basically a technique where by using this dyes, we color the living and dead organisms. So, in staining methods, staining can be two types. One is positive stain and negative stain. In case of positive stain, what happened? Normally, bacterial cell wall are negatively charged, and the stain that whatever we are using in case of positive stain that are positively charged. So, as a result, they will attract to each other. Also mentioned like that. Bacterial cell wall are colored and appear in their background. The positive stain can be divided into two types. One is simple staining and differential staining. In simple staining, we use one type of stain. Either we can use crystal violet, methylene blue, or saffron. In case of differential stain, the name is self-explanatory, where we can differentiate this bacterial cell wall based on their appearance. We use two type of stains. So most common example of differential stain are gram staining and acid fast staining. So in case of negative stain, where the cells remain there, that means that will be uncolored and background is colored to create a contrast, to aid a better visualization of the image. Common example of negative stains are Indian ink and Negrosin dye. So materials required for the stainings are bacterial culture, inoculation loop, slide, normal saline, staining materials, and the last one microscope. So before the start of any staining, first and most important thing is the preparation of bacterial smear. So for the preparation of bacterial smear, first we will take one thin and grease-free slide. Then we put one drop of normal saline using inoculation loop, and after taking the normal saline, we'll sterilize the inoculation loop. After that, after let you have to wait for 30 seconds. After waiting of 30 seconds, you just touch the bacterial colonies. In the touching, whenever you are touching it should remember that we should not scrape the colonies. We just touch the colonies and take it out the little amount of bacteria with the help of the inoculation loop, and in that slide emulsify the bacterial colonies with the normal saline and let it air dry. After the air dry, it should be heat fixed. So in case of simple staining, after the air dry slide, start this slide with the either you can use methylene blue or crystal violet or running for 1 minute. After 1 minute, we wash the smear with the running tap water and let it air dry. After air dry, we can visualize under the microscope. So, in case of simple staining, we can see here in this picture, you can see the diplococci. That means the two bacteria. The two bacteria are staying together. Monococci means one will be one single bacil, one will be single cocci present in the this one's in the slide. And the staphylococcus here the so many coccus in the in your grapes-like structure. Streptococci, there is the cocci will be in chain-like structure. So basically, simple stain we use to see the bacterial morphology and their structure. So next thing is the differential stain. For in the differential stain, the first thing is the gram staining. So gram staining is this one of the most commonly used stain in the microbiological laboratory. Then, after preparation of the smear, we use primary stain that is crystal violet for 1 minute. After the using the crystal violet for 1 minute, we'll just wash off with the running tap water. After the next step, we'll use the mordant. The function of mordant is to fix the color by forming the iodine crystal violet complex. Then after using a mordant, we'll use decolorizer. It is the most crucial step in the gram stain. It will be differentiate bacteria either gram positive or gram negative. After using the decolorizer for 2 to 5 seconds, we use the counter stain or that is safranin. So the principle behind the gram staining is generally bacteria have the two types of cell wall. What is thick cell wall and thin cell wall? So by the using of gram stain, we can differentiate bacteria into majorly into two groups, either gram positive and gram negative based on their bacterial cell wall. In case of gram positive, they have the thick peptidoglycan layer, and in case of gram negative, they have the thin peptidoglycan layer. So we use the in this picture you can see we use the crystal violets for primary stain. Will stain the both of bacteria, either gram positive and gram negative bacteria. And the next step, when you use the mordant, it will in the both bacteria same thing. It will form the crystal violet iodine complex, but differentiate will be in the third step, that is declaring state by the using alcohol or acetone. So basically, what happened in case of gram positive bacteria, due to their they have this thick cellular layer, whatever stain they are using, whatever this after using the stain, they will form one complex. Iodine crystal violet iodine complex. After using the declarator, what happened? The alcohol or acetone, it will be fill up the gaps or pores of the bacteria that they are in the gram positive. So as a result, if the stain goes inside a gram positive cell wall, it will not or it cannot come outside until or unless we use a SIP decolorizer. But in case of thin cell, due to presence of thin cell wall in the gram negative bacteria, what happen when you use decolorizing agent? This is alcohol or acetone. Everything that in this upper layer of the gram negative bacteria, it will be washed off. So as a result, the gram negative bacteria in the decolorizing step, it become colorless. So the last part, when you use counter stain, what happened? The gram positive cellular, they have already took the color of the cell color. They cannot take the more color further. So but in case of gram negative bacteria, they have colorless. Due to that, they will take the color of saffron. So here is the picture of both gram positive bacteria and gram negative bacteria under the microscope. So here you clearly see those who are gram positive. You can see they are violet in color. And in case of gram negative, we can easily see that they are or red in color. So easily you can differentiate the bacteria based on their color. So those who are gram positive or they have a thick cell, or they will take the color of safranin, this blue or violet. In case of gram negative, they will take the color of saffron, that is red. And one thing you have to remember, most crucial step of gram staining is decolorizer. If we use excessive decolorizer, what happen? The gram positive cell will also look like as a gram negative due to excessive use of decolorizer. So we always have to remember that we should not use the excessive decolorizer. In the next the differential stain, next stain is the acid fast staining. Acid fast staining is mainly used for the Microbacterium tuberculosis. That is the main causative agent of TB in the among the Indian population in on all over the world. So, in case of first staining, we also call it gel stain because they developed this stain first developed by Gill and later modified by Nielsen. So we call as the acid fast specialized stain. We also call as a ZN stain. So in case of acetylation, the first I'll start with procedure, then I'll go with the principle. So in case of acid first stain, here also same we are using two type of stain. What is one is carboxyl and that is also called as primary stain. And second, in case of secondary stain or counter stain, we will use the methylene blue. So happen when you after preparation of smear, when you here you have to apply this heat and heat the the lower part of the slide, and in the upper part we have to apply the curl vaccine until or unless and you should remember when you apply the curl vaccine it should not be dry, completely dry. Before drying you have to apply regular in a regular basis. So you have to apply the curl vaccine almost for the 5 minutes until unless their vapor is coming out from the slides. When there is vapor is when you see the vapor is coming out from the slide, you can stop using carbon vaccine. After that, uh, let it cool down for some minute. After the some minute, we will do we'll use the decolorizer. In case of acid we use 20% sulfuric acid. What will happen if your bacteria is a acid bacilli? Whatever stain it has, it will goes inside there. But in case of or in bacteria is non-acidified bacilli, what happened? The whatever the stain in the outside, they take the color of primary stain, that is carboxyl. It will be washed off when you using decolorizer, that is 20% sulfuric acid. So after using 25% sulfur, 20% sulfuric acid, what we will do? We will apply counter stain, that is methylene blue. So what will happen when you apply the methylene blue? Those bacteria who have the those who are bacteria who has the non-acid fast bacilli, they will take the color of methylene blue, which will apply blue in color under the microscope. So principle behind the acid fast bacilli is that those bacteria who have the acid fast bacilli, they have the structure for mycolic acid or lipido material which have the resemble to the wax-like structure. So basically, what happened when you apply the wax, when you apply heat to the wax or wax-like materials, they that time what happened? The wax is start melting. So that time when you apply any type of color or dye, that will be goes inside the bacterial cell wall because they are made that time. So what happened when in case of because basically when you apply the curl vaccine during the heating time, the curl vaccine will goes inside the cell wall or the cytoplasm of the bacterial cell wall. It will be stays there, and when you pull it down, the whatever stains goes inside this lipoid materials that is present in the acid plus bacillus cell wall. It will be stay there forever. So after In the next step, when you use declarator, whatever is the stain, the carboxyl goes inside the bacteria, it will not comes out. So excessive stain will be washed off, but whatever the stain goes inside the bacterial cell, it will be stay inside only. So, but in case of if the suspecting bacteria is non-acidifast bacilli, what happened? The they will not take when you apply the uh, heat, do not take color of colfaxin or they will not go inside. So as a result, what will happen when you apply the decoration? If they present inside the outer layer of the cell also, they will be washed up. So bacteria will be colorless in case of non-acid fast bacilli. So counter stain when you use the methylene blue, they will take the color of methylene blue and you can see the blue in color. So here is the picture of acid bacilli. So here you can clearly see they are almost rod shape and red in color. That is the color of olfactory. So when you see if the if you if someone is suffering or someone is suspecting from uh, TB, so in the laboratory or in the hospital, what will happen? Uh, the doctor will send the patient sample, that is sputum sample, staining or suspecting sample for any other test. So first thing, what in the hospital, what we do? We do acid stain or jet stain for acid fast bacilli, and after the complete the staining, if this person is really suffering from the TB, we'll see some rod-like structure, and it will be in like color in the under the microscope. This is the principle behind, or this is the reason behind being Jaden stain. Next stain is the negative stain. So before I discuss also, negative stain does not stain the bacteria but color the background instead. But the background will be dark and the bacteria will be clear hollow. So we did stain, negative stain. What is the purpose of stain? So it can be used to identify the certain bacteria or structures, that is bacterial spores, and that is very very difficult stain by a normal stain. And whatever we are using, like the methyl, either you can methylene blue, crystal blue, whenever the stains, while using this type of stain, it's very difficult to stain the bacteria is forming spores and any other structure that is normally that we are unable to stain with normal stain we are using. So okay, so generally what happened? The bacterial cell wall is the negatively charged, and the stain we using a negative stain, they is also negatively charged. So as a result, what happened? Instead of attracting each other, uh, because of the both stain and bacterial cell wall are negatively charged, as a result, what will happen? They will repulse to each other. So what will happen? The texture will be, you'll see in the under a microscope, it will be very clear and the background will be color or dark background. So here is the picture you can see that the background, you can see the background is dark and the bacteria whatever we are suspecting they are colorless. So color is hollow in the background. So